Greetings. So let's start with immune cells that are our soldiers. We've talked a little bit about some of the cytokines or chemicals that are involved in immunity. Now we'll talk about cells that are involved in the immunity. So we're going to break it up. The first of these videos would talk about the cells of the innate immunity. So remember, immunity but we divided up into innate and adaptive, and the adaptive we further subdivided it into um, humoral and cell mediated. But today in this video we'll talk about the innate immunity cells. So there's five of them. There's neutrophils, there's basophils and mast cells. So there's neutrophils, there's basophils and mast cells, there's eosinophils, there's macrophages and dendritic cells, and there's T natural killer cells. So we'll try to work through each of them individually so that we have a better understanding of those cells. So the hope is once we understand the chemicals, we understand what the cells are doing, then the next step is to bring it all together in a simplified fashion to share with you what the immune system does and how it acts on different debris or things we don't like. Okay. Neutrophils. So how do we produce it? Leukopoiesis. So remember the myeloid cell line? Migration. How does it go from your blood cells to the site? So I think we've covered that in great detail in one of the previous videos with the cytokines that activate certain pathways, increase the gaps in the blood vessel, and then slow down the neutrophils, hook them in, get them through the gap, and then chemotaxis and attracting them to the actual site. So we've covered all of this stuff. So today we'll talk about, well, how does it act? So here's a neutrophil. So remember, it's got the granules. It's a granulocyte. Remember. So you have the Ben cells, the basophils, the eosinophils, and neutrophils, right? They have the granules, and then it's also a PMN. So it's also its nucleus is multilobulated. Okay. So what does it do? Well, this is my bacteria, and it's got two horns in there, and the horns has an antigen. Okay. So what does it do? Well, it starts to, as it moves towards the bacteria, it starts to extend out its cytoplasm and extend it around the bacteria. Okay. Okay. Until it actually swallows it. Okay. And this reminds me of the movie Aliens. If you've seen Aliens, you had Sigourney Weaver, and there was these machines she used to lift up these heavy containers while they were making their way to the alien planet, right? And so that's the way I think about it. Is it's got these arms, and then it goes around the bacteria, and then it pulls it in and swallows it. So here you have the neutrophil, and it's got the nucleus, right? It's got that bad bacteria, right? And it's actually now uh, swallowed within um, the neutrophil, right? So this neutrophil has several granules. Okay. So these granules actually have the ability to create things called reactive oxygen species or free radicals. Essentially, I consider it like bleach. Okay. So what it does is these granules start to attack this bacteria that is in this area. So there's three things that can happen. First, it dissolves the bacteria. And then once it dissolves the bacteria, You only have the antigens or the surface markers or the different molecules in the bacteria that remain. They're basically antigens. So again, this was the area where the bacteria was swallowed, the granules attacked it. Now you only have antigens that are there, and then this makes its way to the surface, and then it gets released. Okay? And we'll talk about what happens once it gets released, but it essentially releases antigens because the bacteria is no longer there. Bacteria is gone. Okay? What else can happen? Well, if it's a really tough bacteria, we've got a problem. It might take lots of granules to start to hit that bacteria and try to dissolve it. Well, all of this bleach as it comes out is going to actually damage the neutrophil, and the neutrophil is going to self-destruct, and that's called oxidative burst. Okay, and all that bleach that's released also gets out and tries to damage the other buddies that this bad bacteria may have had. And the way I think about it is it's a self-destruct mechanism, especially when the bacteria is tough. So remember the movie Predator? So at the end of that movie, you had Predator that met very difficult, tough Arnold Schwarzenegger. So what it did was it activated the self-destruct button, and we had the activated burst, and it tried to take out everything and everybody, right? So that's the second thing that it can do. So it can dissolve the bacteria, and again, if bacteria is not very hardy, the antigens get released. If the bacteria is very hardy, then lots of bleach, which destroys the cell. Again, the antigens come out. Okay, so that's two. Well, there's a third possibility as well, which is if the bacteria is still very tough and not responding, what happens is the neutrophil is very smart. It can try to net it. What does net it mean? Well, it uses this coiled DNA that it has, right? The DNA is coiled, and uses it as a net to trap the antigens and the bacteria. Okay? And now that it's trapped in the extracellular space, it gives the immune system a chance to respond to it. Okay? And so it nets it. So the neutrophils use their DNA as trap in extracellular space. So neutrophil, extracellular, traps, nets. So that's what it does. Invokes an immune response and kills the hardy bug. Okay. Here the DNA though usually is intracellular inside the cell. This DNA now comes out. And so there is a potential that the immune system will tag this genetic material as enemy along with the antigen on the foreign molecule. Okay? Just something to think about. But essentially, that's how neutrophil works. Okay? Neutrophil has three mechanisms. Okay? It basically phagocytoses or eats up or swallows the bug and attacks it with bleach and then releases the antigen. If it's tough bacteria, it can self-destruct or net it. But either way, the result is you have antigens that get released, okay? And they go into the lymph system. So we'll talk more about the lymph system and what happens to it a little bit later. Let's go to the next one, which is basophils and mast cells. So we've talked about how they're created. So again, if you're not sure, go back to the anatomy and physiology video, which talks about leukopoiesis. We've also talked about how they migrate. The big way that they act is the antibody system, where IgE antibodies get produced with first exposure to the foreign molecule, and then IgE attaches to the mast cell, and once re-exposed, mast cells degranulate, causing inflammation. Okay, and then phagocytosis of the foreign material and its removal. Okay, so that's how basophils and mast cells act. So the big thing is again their role with histamine release and foreign molecules and allergy. Okay. Eosinophils, again, we talked about how they were created and how they migrated, right? So they're one of the Ben cells. Okay. So we talked about them. We talked about the myeloid line. If you're not sure, make sure you review the video. Again, their role is in parasites and worms. And again, you have the antibody IgE that tags the worms or parasite. And then the eosinophil attaches to the IgE and gets activated and releases toxic proteins that kill the worm. Okay? So that's how eosinophils act. Okay? So those are the three cells. So let's continue our journey into macrophages. Okay, let's talk a little bit about macrophages and dendritic cells. Okay, so again, so remember when we are looking at how they're formed, we've already done a video on it, right? So take a look at it if you're not sure about it. Okay, which line do they belong to? Okay. Additionally, are they granulocytes or agranulocytes? They don't have granules, right? Are they myeloid or lymphoid? They're myeloid line. Okay. Additionally, they're also antigen-presenting cells. Remember these and the B cell lymphocytes are antigen-presenting cells. They have that MHC2 molecule on their surface, which they use to present the antigen to another part of our immune system. Okay. So now that we have recollected some information, let's just focus on macrophages and dendritic cells. So macro means big, phage means eater, big eaters. So it tells you what it does. Okay, how do they migrate? We've covered that as well in terms of cytokines and how they cause inflammation, make the blood vessels leaky, and then through certain processes, slow them down, get them through the gap, and then chemotaxis or get them to where um, they need to go. Okay. We've also talked about interleukin 1, right? And what it does in terms of inflammation, and it gets more of its friends. Okay. So we've talked a little bit more about it. Okay. So how does it work? How does it work? So there's a couple of pathways that it can take. Okay. So again, one is very similar to neutrophils of how it gets swallowed. The other pathway is, if you remember from the complement system, there's an alternate pathway where the macrophages can attach directly to the antigen on the bacteria through the CB receptor. Remember that? That's the alternate pathway. The result is the same. It gets swallowed. Okay? And then you have lysosomes that act on this bacteria that's swallowed. Okay? And then it gives you antigens. Now these antigens, remember neutrophils, these antigens got released? Well, in macrophages, they don't. These antigens are processed by the cell, and they're put on an MHC2 presenter complex. So you have the MHC2 presenter complex that gets formed, and then you have these are the antigens. So there's one antigen per MHC2, and that actually gets to the surface. So essentially, you have this antigen presenting cell, that now presents the antigen on the MHC2 complex, which is the foreign antigen. Now remember, these are special cells. They have MHC1 and MHC2. Most of the other nucleated cells have MHC1 as well. Remember MHC1? That has the self antigen on it. Okay? So now this big molecule goes into the lymph channel, okay, to be processed. So there's two things that go into the lymph channel. You have the antigens that can go in, or you have um, the antigen presenting cells that are presenting antigens that go into the lymph channels. Okay, and that's macrophages and dendritic cells. What do? They present this antigen to T helper cells in the adaptive immunity. So we'll talk a little bit more about it when we talk about adaptive immunity and more specifically humoral immunity. So again, this is external extracellular threat that gets processed this pathway. Okay. Now we have another cell. So we're going to go to the next cell line, which is our T natural killer cells, or TNK. Okay. So let's talk about is this an innate immune cell or adaptive immune cell? So it is innate immune cell. It's a T lymphocyte. Okay. So again, it's a lymphoid line. So this is where it becomes very tricky. So remember, lymphoid line gives you B cells and T cells, and then T cells give you T cytotoxic cells, T helper cells, T natural killer cells. B cells, TC, cytotoxic cells, and helper cells are all adaptive immunity cells. This is the only lymphoid line cell that is innate. That's something interesting about it. Okay. Now, this natural killer cell does not manage extracellular threats like bacteria. It manages endogenous intracellular threats such as viruses or tumors. So what does that mean? That means so viruses have to get inside the cell, integrate into the machinery of the cell, and only then they are able to function and do the damage. Versus bacteria that are alive and don't need to go inside the cell to get revved up and going. So T natural killer cells are cells that target viruses and cancer that are problems which are within the cell. Okay. Um, so we talked about how it's created a part of the lymphoid line, and the next thing is how does it get to the site? So I think we talked about interferons alpha and beta. So remember when a cell is infected with viruses, one of the things that it does is it lets the other cells know, look, there's interferon, that something's wrong with me, and that you guys get ready. And then they do it through interferons alpha and beta. And the alpha and beta does two things. First, goes to the nearby cells and creates these scissors, protein kinases. The other thing it does is it attracts these natural killer cells. So let's say the natural killer cells are now face to face with a bad cell. This is my bad cell. Okay. Now my bad cell, so my normal cells are going to have an MHC1, right? And that's going to be attached with the self antigen. Okay. Now, let's say a virus attaches itself in my machinery, or a cell becomes cancerous. What may happen? It may completely lose its MHC1. If that be the case, natural killer cell will recognize and say, this cell's no good now that it's there, right? What else can it do? It can change that MHC1 cell. So instead of the self antigen, it's going to have something extra with the self antigen. And natural killer cell is going to say, not good. Or this bad cell may have a very specific type of antigen, and the IgG antibody would be attached or would tag this bad cell. So remember the antibodies. We've talked about this for a little bit, and we have a video where we talk about it in detail. But essentially, our luggage is tagged, and that helps us flag this cell to be a bad guy. Okay, and then the natural killer cell, once it recognizes the bad guy, so it comes to the site with interferons alpha and beta. Through these three mechanisms, recognizes that a cell is not good, and then it creates proteins that creates that hole in that bad cell, or starts the process where the bad cell auto destructs or goes through apoptosis, and that's called the self-destruction of the cell. Okay, and that leads to death of the cell with viruses, or if it's a tumor cell. Okay, so that's sort of the overview of our cells. Let's go through it one more time, just to recap real quick. So again, we talked in this video about the cells of innate immunity. So we talked about neutrophils, basophils and mast cell, eosinophils, macrophages, and T natural killer cells. With neutrophils, we talked about how it phagocytizes or eats or swallows the bugs, and then it releases the antigen, and it does it through three mechanisms. It dissolves it through granules, it can self-destruct with oxidative bursts, or it can net it. And the end result is antigen that goes into the lymph channels. Basophils and mast cells are big on allergy and autoimmunity, allergies. And again, that's more of the histamine and how that degranulates when the foreign molecules are there. Eosinophils are cells that attack the parasites or worms that are generally tagged by IgE, and so they have a role to play with parasitic infections. Okay, so those were very quickly the three that we covered. Then we talked a little bit more about the macrophages and how it swallows, yes, the bad guy, but through two pathways: CB receptors using the complement, we've covered that, or swallowing it, and then it actually takes the antigen but doesn't release the antigen like neutrophil. It actually presents it on the MHC2 molecule on its surface, and then goes into the lymph channel, and then presents the antigen to the T helper cells. And then last but not the least, we talked about the natural killer cells, which get attracted to the site by interferons alpha and beta, which is released by cells that got infected with viruses or tumor cells. And then through, once it's close to the bad cell, it checks out and sees if the cell has no MHC or an MHC1 that has changed or is tagged with an IgG. And if it is, it gets activated, creates hole in the bad cell, or also starts the process of apoptosis, which is self-destruction of the cell, both of which leads to the destruction of the cell with viruses or a tumor cell.